G'day, Dr. Carl, University of Sydney, Shirtloads of Science, today with, now what's the correct pronunciation, Renee Goreham?
Oh, Goreham.
Goreham.
It's a silent H.
Oh, so there's a silent H. So Renee Goreham, and you're a physicist?
Physicist, yeah.
And you have been able, in two separate thingies, on one hand, as a physicist, find cancer, signs of lung cancer in the air that people breathe, and also use physics to image body tissues with red light, which could cover anything from hand injuries to breast cancer. But you're a physicist.
Yes.
And you've ended up in the life sciences.
Yeah, so it's often a running joke that I'll say extracellular vesicle is short, the short version is EV, and people go, oh, electric vehicle? But I haven't found evidence of lung cancer in breath yet, but that's the plan. That's a blue sky. So
you're heading down that pathway. So let's just sort of go down that pathway. And, diagnostics are a good thing to have. Yeah. So you're a physicist, and you're I'm guessing you're not a theoretician, you're a...
experimentalist.
Okay, so just for the audience who don't know, by the way, audience, apparently there was a famous Nobel Prize speech where the winner in physics said, I'm an experimentalist and thank you for the Nobel Prize, and if I've failed to thank any theoreticians, it's not because I've forgotten to do so, but because I hate them. Because they come up with an idea when they have a cup of coffee, and I have to spend 10 years working it out, and then even before I've got started, they've had another cup of coffee and they come out with more ideas, and they just increase our workload. So I'm not going to thank any theoreticians. So there. But so do you have any theoretician friends?
Yeah, so my office neighbour, Karen Livesey, I respect you, and you're a great theoretical physicist. Yeah, she's my office neighbour, and she's the polar opposite to me. So we both do nanotechnology in some capacity. I do it in the lab, and she does it with pen and paper. Yeah.
Right. So before we dive into that, the nanotechnology means really small, and nano is literally a billionth, so we're talking very small scale. So just for the audience, as a real physicist, which I'm not, can you explain what's the difference between a theoretical and an experimental physicist? I was told the difference is that when a theoretical physicist walks into the photocopy room, the photocopiers die.
Yeah. That's probably true, actually. So I guess a theoretical physicist would predict things based on mathematics and maybe do some computer modelling to back that up, whereas I use all my applications in the lab. So my biggest thing that I have to do as a nanotechnologist is prove that what we can't see is there. So I use an array of experiments to prove that they're there and they exist and they do the things that we say they do. Yeah.
How weird. So you're like these wizards working with invisible things saying "badum," and therefore, and you may or may not have lung cancer. So take us down this pathway and you're heading down this pathway still of trying to have a nice screening test for lung cancer based on breath, because normally it's diagnosed when it's too late. Hey, doc, I'm having trouble breathing, blah, blah, blah. Okay, it's well and truly advanced, whereas you want to get in the early stages.
Yeah.
And I was reading your fine papers here. Isolation, characterization, and detection of breath-derived extracellular vesicles.
Yeah.
Now, should we describe cells just for the audience in case they're geologists and they don't know what cells are?
Yeah. Well, cells are everything sort of living in your body that it is like a skin cell or a lung cancer cell, or you've got the stem cell that can turn into any of those. And each cell will release these vesicles, extracellular vesicles.
Really?
Yeah. And they almost mimic what the cell is doing or what state it's in.
Really?
Yeah.
I didn't learn this in biology. Like, I learned that there's 37 trillion cells in your body, and the majority, 2/3, are red blood cells. And we learned that there are various biological machines inside. Like, you got a membrane, and you got the nucleus, which kind of tells you what to do, kind of. And then you got Golgi bodies and other things in there. And then we saw vesicles, but these vesicles they're little balls, I'm guessing, inside the cell.
Yeah.
And the cell would be how big? Maybe 10 millionths of a metre across?
Yeah, it depends on the cell, but it could be about 30 microns.
30?
Yeah,
a micron is a millionth of a metre. Yeah,
yep, that's right. You're very good with that. I'm good with my small sizes. I can't go back up. Yes.
You only deal with the really small stuff.
Yeah.
Okay.
Yeah. So cells will produce these vesicles, and it was back in the 80s, I believe, that they were first discovered, and they were always thought to be trash compartments. So the cells produce these little bubbles that with a biolipid membrane, just like the cell, when they have proteins embedded with cargo of DNA, protein, and RNA. And they just thought it was trash. But it wasn't until later in the 2000s or maybe the late 90s that they found that they actually have roles. So it's not just cells getting rid of their trash, it's actually the cells communicating or starting processes in the body, a whole array of different things.
Okay, now you mentioned biolipid membranes, so you've got the cell like a basketball, and then inside you might have something, say, like a marble, and it's roundish, a ballish, and the membrane to isolate it is fat and then something else in fat. And is that right? That's what biolipid means. And the advantage of fat is that it sort of resists the water, so it doesn't get dissolved by the water in the cell, so it can stay as its own structural, have its own structural integrity. Is that...
Definitely, yeah. I've always been fascinated by lipids as well because they do form the barrier for all cells.
Oh, really?
Yes.
They do, don't they?
Yeah.
I never thought about that.
But they're actually quite fluid. So I'm a physicist, so I think they're just forming like a fence. And so the fence would have two bits of wood, two heads that like water, and then the tails that form that lipid that hate water. But in the cell itself, it's not rigid. It flows and the proteins move around. It's almost like a water. But that's really hard for me to comprehend, and I guess that's why I like it.
So you got these what's the proper pronunciation? Vesicles or blebs? Were they called blebs once upon a time? Or
they bleb from the cell. So the scientific term for when a vesicle is released from the cell is blebbing. Yeah.
So you've got this big ball, like a basketball, and inside you've got a little marble. And what the marble kisses up against the inside wall of the basketball, and then what happens?
Well, yeah, so there's two mechanisms. One that I don't care about that much is just general vesicles. They're larger in size. They're not as interesting.
Yep.
And they just form when the membrane just blebs a bubble out. And so it just takes some of the inside and expels it.
So the ball comes up and then kind of opens up and then joins with the basketball membrane or skin, and then that opens up and then it just... Does the vesicle itself go through the basketball? Does the marble
it's formed from the membrane itself.
So it's a dynamic thing.
Yeah.
So it coalesces and then reforms itself.
Yeah, so I guess that's why they called it blebbing. But yeah, the ones that I'm interested in are the ones that are formed in a different vesicle. So there's all different types of vesicles, and there's a micro...
What was I told? Why did we get the email? Okay.
Yeah, and the microvesicles inside the cell are formed in a different pathway. It's quite complex. And the things inside used to be called exosomes.
I remember those names.
Yeah, but now they've changed the name of them. But it makes it hard because now everything's called extracellular vesicle. But
so what do they change it to?
Small extracellular vesicle.
Right. And so you've got this vesicle inside, and then a little bit later it's outside the cell.
Yeah.
So that means that it might be in the connective tissue. So what's the... And before we get to how it gets the breath... What did they think was in it? You said proteins and DNA?
Yeah, proteins, DNA and RNA. So what I've been able to do is image these with an electron microscope because they're so small. You can't use a confocal or a light microscope. You have to use electrons. And so you can image and you can see the biolipid layer with those lipids in the heads and stuff inside.
Wow. So how big are these vesicles? Are they... like a micron across or a millionth of a metre?
Oh, it's 50 nanometres. So
50 billionths of a metre.
Yeah.
So visible light is 400 to 700 nanometres.
Yeah.
And this is way below the blue. It's 50 nanometres.
Yeah. And I guess
that's really small.
Yeah. And that's what sparked my interest because I like nanotechnology. I make nanoparticles in all capacities. And I thought, what? There's naturally occurring nanoparticles. And they're useful for detecting different diseases.
Right, so we're getting to that now.
Yeah.
So the vesicles are about 50 nanometres across, are they? Mm-hmm. So you'll publish a paper and it'll have a picture of a vesicle and then there'll be a bar that says 50 nanometres or 10 nanometres, giving you the scale of it.
Yeah, there'll be one in that one actually.
That's how I was stealing that interpretation. Yeah. And then, but if it's got proteins and DNA and mRNA, then it's probably not just waste products because they're useful things, aren't they?
Yeah, exactly. And that's what was later discovered, that they're useful. Also, the composition strongly relates to what's going on with the cell. So
so they reflect, they're like a mirror of the activity of the cell from which they left.
yeah, I call them
Really?
like little cell mini-me's almost.
Mini-me's?
Yeah.
Nice.
Yeah.
Nice.
So, yeah.
And so, what do we think why does a cell do this? Why does a cell just decide, hey, I'm a lung cell or a liver or a thymus cell, and I'm just going to squirt out this vesicle and bleb it out into the surrounding tissue around me? Why does it do it?
Well, yeah, that was the first question. And I guess there's many different roles they play. So the ones that I know of is just talking to other cells.
Talking to other cells. Because our body is not just a bunch of individual things. It's a composite that talks to each other inside.
Yeah, there's a big group of researchers at La Trobe Uni, and they were looking at the role of extracellular vesicles in prion diseases. And you could actually get cells to be infected or changed to have prion characteristics based on just them uptaking those extracellular vesicles.
Wow. So you've raised a new issue, so we have to define a prion.
Yeah.
So a prion is a protein that is possibly a bad protein and it's misshapen, and proteins do their things, at least on one hand, because of their shape. And when a prion comes up against a regular protein that has the same atoms in it, same number of atoms, it makes it change shape into the shape of the evil prion. Is that how it goes?
Yeah, I'm not an expert on definitely brain diseases, but something like that.
Jakob-Creutzfeldt disease and mad cow disease and stuff.
Yeah.
So what was the thing you were saying about the prions again?
Saying that the extracellular vesicles that came from prion-diseased cells...
From cells that had already been infected by prion disease, like mad cow disease?
Could potentially infect other healthy cells, but it was done in cell culture, not in clinical. But so there was that evidence. That was probably back when I was in New Zealand. So I think 2010. No, I don't know, 2018. Yeah, a while ago. Yeah.
So we've got these vesicles being squirted out into the intracellular or the extracellular tissue, and then do they make their way into blood vessels? Then eventually we're heading towards the lungs and the lung cancer thing. So take us down that pathway.
Yeah, so a lot of research is looking at how we could get these extracellular vesicles from blood. So
they will end up in the bloodstream in some cases.
In fact, in almost every bodily fluid, you'll have these extracellular vesicles. So, urine, saliva, blood, breast milk. I did some research when I was in New Zealand on that. So, they're released everywhere.
And they're possibly to do with communication, and I guess they make their way into the red blood, into the blood vessels through the capillaries where there's little gaps in the walls.
They're really stable. So there's two options that people are researching, one for disease detection, or there might be more, but these are the main ones, and then one for drug delivery. So you could potentially use these. And I did a little fun project as a physicist. I zapped them and loaded nanoparticles inside. That was fun. So, they're the two avenues that people look at, drug delivery or disease diagnosis.
Okay, so getting back to your situation, so you've got the lungs, and that's where gas exchange happens. You breathe in air, forget the nitrogen, it's got 20% oxygen and there's a bit of gas exchange, and then you breathe out 16% oxygen and 4% carbon dioxide, and the gas exchange happens over an area of, I think, of about 70 square metres, which is like half a singles court in tennis, so it's a huge area. So now take us to the lung cancers, and there's different types of lung cancers, we won't go into that, and you're looking at some that are not the small cells cancers, so take us through that then.
Yeah, well, we haven't done too much on lung cancers just yet. So the first step was we don't actually know where the extracellular vesicles are coming from. So we've so the idea was we saw that there was evidence that these were in our breath, these extracellular vesicles. No one had done that. Well, proteins. So
were you involved with finding them in your breath?
Yeah, yes. So a master's student, Garima, she saw this paper and she's like, there's evidence that there's biomarkers specific to extracellular vesicles because there's some proteins that exist in these extracellular vesicles that tell us that they are those things. And so she found this paper and I was like, okay, well, I can't ethically ask you to give me your breath. So I went into the freezer.
You went into the freezer?
Yeah.
What do you mean the freezer? Like you mean you got a walk-in freezer, not just you didn't do some gymnastics and fall into a really small freezer?
We were lucky enough to have a walk-in freezer, otherwise that would be awkward. Went into the freezer, blew into a tube and condensed the water vapour in my breath to see if we could find them.
Why go into a freezer? Why don't you just do it out in the open? Because you want to see where the breath was going.
Yeah, and we wanted to condense it into a liquid form to collect it.
And you want to condense it immediately on the spot.
Yeah.
Good thing you did to yourself. You didn't have to go through a whole ethics committee. I want to walk into a freezer, and then 15 years later you're allowed to do it.
We have a very much more sophisticated, well, I would say sophisticated, partly sophisticated mechanism to collect breath now, where you have a furnace which we put in the freezer, and there's a collection tube, so people just blow into this tube. And we're able to collect their breaths, and that's with ethics.
So then you walked into the freezer, you collected some liquid, and then?
Yeah, then we isolated them. So we removed all the unwanted stuff. We removed the proteins that are in there, all the other stuff that's in your breath, and we're able to.
There's proteins in your breath?
Oh, well, yeah, there's all sorts. Anything biological could be in your breath. I haven't characterised it, but other people have. It's quite complex.
Wow.
Yeah, but if you think about blood, that's also complex, and you can isolate it using the same method. So you get the pure extracellular vesicles, or as pure as we can get, and then we can image them, use protein analysis to make sure they are there. And, that's what we reported for the first time in that report.
So because you're getting breath which comes from the lungs, if you've got a pathology growing in the lungs, like a lung cancer of some sort, you might be able to then get vesicles from that going into the breath, and then maybe you're heading for like trying to develop an early screening test? Yeah,
so that's the plan. So another student, Emma, there's been 3 students. So it was started with Garima, went to Zarina, Dr. Zarina now, and now Emma, who will be hopefully a doctor next year. And she's the one that's now trying to go to the next step. So we've collected a whole heap of healthy cells, extracellular vesicle breath, and we're trying to find out where those come from. What cells? Do they come from the lung, or do they come from the airway? And then part of that work is also to move on to collecting breath samples from lung cancer patients.
Right, so that's where we're heading for. So how did you get to this stage where you end up doing these weird things of walking into a freezer and look for things that are too small to see? So I imagine you started off at primary school and high school in Australia, was it? Yeah,
I grew up in the country of South Australia, so I moved around a lot because my dad was in the mines, and we just moved around a lot. I went to high school in Port Augusta. So if anyone knows that town, it's quite small.
The point of the Spencer Gulf there?
Yeah, in South Australia. And so from there I was like, I could be a pilot, or I could be something. And I just went to science because that's what I was good at.
And you managed to avoid getting lead poisoned?
Yeah, I did. Well, that I know of.
Yeah, there's a bit of a problem with that in some parts of Australia, including Port Augusta. They had
Port Pirie, yeah.
Port Pirie. Okay,
yeah.
Okay. So
that was very close.
Okay, that was a detour. Forget that one. So then you decided to, after school, go to
university to study first civil aviation, but then I realised I really liked the physics part.
Oh, really?
And I was too poor to fly.
Because you need to spend lots of money, like hundreds of dollars per hour, to get a whole bunch of hours up.
Exactly, and at that time there's no way I could afford that. So I went into science, and I think it was my last year I did honours, and I did it in nanotechnology, and that's when I fell in love with the research component. So doing things that haven't been done before, like walking into a freezer, collecting my breath sample and proving that there's extracellular vesicles there.
Because that's the weird thing about science, is that you discover something that nobody in the entire known universe has discovered, and you're the first and only person to know it for a little while.
Yeah, that's right.
Wow.
Yeah, and now going to other conferences and seeing other academics going, look, breath is a great source. It's validation as well. So.
No, but how did you end up from that into Newcastle Uni in physics?
So I went from Uni SA to Flinders Uni, undergrad and honours, and then I went to Uni SA to do my PhD again. And it was there where I had a nanotechnology project where I was making nanoparticles using a plasma reactor and then growing cells on them. And I'd never grown cells in my life. So Yeah.
Well, you're a physicist. What would you want to do with this squishy stuff?
Yeah, so I learnt that whole new thing in my PhD, and so that's where I sort of started to spread and become multidisciplinary, as they say. Yeah.
Wow. So the thing I can understand working here in physics is that physics is sort of a preparation for problem solving in any field, which on one hand can lead to a sort of casual arrogance of I know everything, but at least you know the basics of how to do experimental work.
Yeah, and I think if there's anything I don't know, I have the resources to find out how and to do it.
Sure.
Yeah.
You search the peer-reviewed literature. So that's one pathway that you've been going down. And then also you've been doing something with red light to look into and through body tissues to diagnose again. Is that for diagnosis again?
Yeah. I have another student, Kathy, and a colleague, John. So they both work at University of Newcastle.
Yeah.
And what she's done is she's built a system where it uses red laser, shines into a gelatin jelly sample, and able to image what's inside by capturing the image of the scattered light. So it goes in and then the sample and then gets scattered, and we can take a picture and then draw back, almost back project what caused it to scatter and recreate an image.
Is the word convolution involved in there somewhere?
Yeah, probably.
Right. So in that case, you're coming up with a potential body imaging modality that doesn't involve X-rays.
Yeah.
And so, for example, in the case of breast cancer, 1 in 8 women will get it, 1 in 40 will die. And if we can diagnose it earlier and certainly not put X-rays in, that's all to the good.
Yeah.
So that's another thing that you as a physicist are doing.
Yeah. So we've just putting the paper through peer review at the moment. But yeah, it's really good because it's not something. I mean, everyone's probably shone a torch through their hand and seen the red diffused light. So it means it scatters everywhere and it becomes larger.
Yeah.
And you see that red colour. So being able to use that, take an image and then decide what's inside is actually quite unique. And so that's where the novelty comes. So we could replace the need for squishing of the breast tissue and also the ionising radiation that damages some of the cells in some capacity.
But being very humble, you neglected to point out that you're one of the 60 stars of STEM here in Australia.
Yeah. Oh, yes.
Can you tell us about that?
Yeah. So the Science Technology Australia, they have this program that goes for 2 years. And so 60 people from across Australia in all capacities of science, in industry, university, everywhere, get chosen to become superstars of STEM for 2 years. And so we go through different professional development on media skills, communication, sharing our love for science, which is something that I've always been passionate about, and then even meeting up with politicians. So it's got all avenues of communication.
So how far are you through this 2-year window?
Oh, yeah, actually, thinking about it, I started in December. And so, yeah, probably, 9 months now. And so, yeah, it's been really great. We had a meeting this week even, and it's just really unique to have people, women, 60 of yous, all coming together to talk about your science and just be really supportive. It's a brand new experience, but I'm loving it.
And also build up the relationships for the future.
Yeah.
So you're a really good example of somebody who started off here and then ended up, God knows how come, looking at microscopic things you can't see with the human eye, and in some way further down the line that will better diagnostics for the human race.
Yeah. In fact, it's a funny story. My mum was telling everyone in Port Augusta that I'm a famous forensic scientist because she can't say physics. Sorry, Mum.
Close enough.
Yeah, close enough. And I appreciate that she's proud of me. So.
Now, I noticed before when you talk about your students with whom you worked, they all had names ending with A.
Oh, I didn't notice that.
Does that have to happen? So tell us, do you take students and do they have to have names ending in A, or any name will do?
Yeah, no, any name will do. Anyone with a passion for science. That's one of the parts of my job that I love is mentoring the new generation of students and researchers. So, yeah, I have 7 PhD students.
7?
But
what a workload.
Yeah, but 3 of them have completed this year. So I have 2.5 doctors. One will become a doctor in the coming months.
Wow.
Just this year alone. And that's probably the highlight of my career to date is seeing those students progress to finish.
Wow. So part of being a scientist is that you train other scientists along the pathway. So how do people find you besides looking up for Renee, R-E-N-E-E, Gore, no H, you don't pronounce the H, Goreham, but G-O-R-E-H for hotel, but you don't pronounce a hotel, A-M. So they can find you at Uni of Newcastle in Australia. How else can people follow you and your fine work? And
I'm one of the few academics with TikTok, so you can find me. Dr. Renee Goreham on TikTok and Instagram. Also LinkedIn. I've had people contact me there, or email. I'm really responsive to emails. So, all of those factors. I'll have a website soon, but that's in the making.
Wow! Look, thank you so much, and thank you for taking us along the pathways. It's gone from there to here, and who knows where it'll go in the future.
Well, thank you. I've been a long time listener, so I was very excited to be part of it.
Dr. Renee, thank you.
Thank you.
Shirtloads of Science is washed, spun and aired by the University of Sydney.